Hello, good afternoon. So, good afternoon, friends. Now, today we'll discuss about the structure of the gene, regulation of the gene, and the meaning of biotechnology, recombinant technology, and genetic engineering, and their applications. Now, to begin with, first of all, let us know a bit more about the gene, concept of the gene. Before knowing how a gene works, how the work of a gene is being regulated by other factors. Now, gene, the concept of gene and the meaning of the gene has been changing from time to time. And, it was Johansson who first proposed this term gene. And it was, Mendel, by performing various experiments in Pisum sativum, that is garden pea, and proposing various laws of heredity long ago. And in 1860s, but of course, as you know, it remained unnoticed for almost 35 years. Later on, it was in 1900, three people, it was Devries, Correns, and Teschermark, who made an attempt to rediscover the laws proposed by Mendel. And thereafter, the Mendel's work was reinvestigated, and more and more knowledge in genetics is being added. And because of the pioneering work taken up by Mendel, we call Mendel as the father of genetics. Now, during his time, the term gene was rather unknown, and he called these, units, the units that get transmitted from one generation to other generation, and that are responsible for the phenotypic expression and the genotypic variation, and the some characters that are being expressed, some are being hidden. And based on all these variations or changes he could observe, he proposed various laws, and thus he is rightly considered as father of genetics. And he called these units as factors. And in modern sense, the factors are equivalent to our what we call genes. Now, gene, as you know, it is what we call as an atom to the matter. The gene is to what we call a biological activity or a living being. Now, it is the unit of what we call transmission. It is the unit of heredity, and also it is the unit of function. Because it is the unit of transmission, why it is known as unit of transmission? Because it transfers characters from one generation to other generation. So that's why it is known as unit of transmission. And these characters that are being transmitted are in true cells. That is, the offsprings or the daughter cells or the progeny resembles their parents, because the characters that are being transferred have a hereditary bearing. So that's why the genes are units of heredity. Now, they are also otherwise called as units of function, because, of course, the ultimate expression of the gene is in terms of function. It has a function to perform, maybe in terms of production of a protein, in the production of a hormone, in the production of a vitamin, and a production of enzyme. So almost all the biological activities or physiological activities that do occur in a cell or cytoplasm are directly or indirectly under the control of the gene. Now, this gene, which was rather a particle in good olden days, it was considered as a particle as such. Later on, it is now, it is a part, a fragment. A fragment of a DNA is a gene. A fragment of DNA that can code for a polypeptide chain. That polypeptide chain is nothing but what we call a cistron. That is a gene. Now, gene, as you know, it is liable for change. This change we call a mutation. Of course, yesterday we discussed about various types of mutations, the point mutations and the chromosomal aberrations, etc. Wherein, so the gene is subjected to variation or change that we call a mutation. So as a result of mutation, the gene gives rise to an alternate of the original one. That alternative character is what we call an allele. So that's why a gene may have one or more alleles, the alternative forms of a gene, they are the alleles. So a gene could be an allele. Then gene, as I told you, gene can be what we call a fragment that can code for a polypeptide chain, that is in terms of a mRNA. That can also be called as a gene. So here, up to almost 60s, people thought that gene is the smallest unit or an indivisible unit. It is a unit of structure, it's a unit of function that can't be further divided. But it was due to the brilliant works of so many people like Benjer. He said that gene is divisible. He says that gene can be divided into two subtypes or subfragments. They are what we call a cistron, a recon, a muton. What is a cistron? A fragment of the DNA, a fragment of the DNA, a part of the DNA, or a part of the DNA that has hundreds or thousands of nucleotides, can code for an mRNA, and in terms code for a polypeptide chain. That can be a gene. So he says that, Benjer says that gene can be divisible into what we call a cistron. Cistron is which can code for a polypeptide chain. Then, not only that, even a cistron can be further divisible. It may consist of a couple of nitrogen bases or more than that. That can undergo what we call recombination, a unit of recombination. So that's why he called it as what we call a recon. A cistron, a recon, and the smallest, of course, maybe just a pair of nucleotides. That is a pair of nitrogen bases. That is what we call a muton. Unit of mutation is a muton. Unit of recombination is a recon. So what I mean to say here is, gene is a fragment of DNA that can undergo a change in terms of a muton or a recon or code for a polypeptide chain in the form of what we call a cistron. So this is a gene. So gene consists, so the number of nucleotides in a gene or the number of genes in an organism varies. For example, if you take E. coli, Escherichia coli, the number of genes in them, in E. coli, are estimated to be around 3,000. And the number in our human beings will be more than, or they'll be in thousands. And the number of nucleotides will be in billions as such. So the size of, or the size of the DNA, or the number of nucleotides, or the number of genes vary from organism to organism. They vary from organism to organism. So here, this gene, of course, genes are there and they are responsible for transmission of characters. They are responsible, or they do undergo change in terms of mutation. Okay, now these genes, they directly or indirectly determine all the biological activities, all the physiological activities that occur in the cytoplasm. Now, the expression of the gene or the regulation of the gene, the expression or regulation of, so it was, Jacob and Monod. They were the people, Jacob and Monod, they proposed what we call one gene and one enzyme. So one gene and one enzyme, of course, one gene and one enzyme, it was not because of Jacob and Monod. Jacob and Monod, they proposed what we call gene regulation or lac operon. And the other people, many people like Benger and others, they have contributed a lot to our knowledge of gene. Now, this gene, how it expresses, the expression of gene. So here expression means the functional part of the gene, the functional part of the gene. It was the brilliant work of Jacob and Monod. Based on their work, they proposed a concept of what we call operon concept. This operon concept says that this operon is nothing but a combination. This operon is nothing but a combination, a group of closely placed structural genes and regulatory sequences, regulatory elements or sequences of the DNA consisting of regulator, promoter, and operator. That forms what we call an operon. So operon is nothing but a group of closely placed genes. Now these genes, for our convenience, they can be divisible into what we call structural genes. They are the functional, structural and functional genes, and the other is what we call control genes. The other is control genes. So some are, structural genes, the other are control genes. Now, if you take the operon concept given by Jacob and Monod, and just before discussing this operon concept, this every cell tries to save its resources, its energy. We face lot number of constraints, maybe in the form of availability of the resources, or for example, if you take the petroleum resources, now we have to import them. So that's why the government and other environmentalists, they stress that they have to be judiciously used. They should not be misused. Now that leads to what we call inflation, and also it spoils your environment. So they have to be judiciously used. So that means we have to use vehicle or these energy giving substances only when it is absolute. When it is absolutely necessary, then only we should use. So here also genes are there. Genes are here. They use the ATP or the energy. So for every function to be performed, it needs energy. Now here, these genes, they should not be in an active state always. There is a mechanism to control the energy, so wherein the genes are either switched on or off. So switching on and off of the genes is in turn controlled by the substances that are present in the cell vicinity, that is in the cytoplasm. So when it is necessary, then only the genes will be switched on, or else they remain, inactive or in off form, that is switched off. Say, for example, we have current. Now this only when there is darkness, when it is invisible, then only you should put on your light. If it is not needed, then you should put it off. So also, thereby you are saving the power. Thereby you are saving your electricity bill. So here also, a cell, if you take a cell as a unit, it also has its energy requirements. It has, genes. So they are to be used judiciously. So for this, there is what we call a control mechanism prevailing. Now this control, whether to a particular gene has to be put on or switched on or off, is under the control of what we call control genes. These control genes are, say, for example, regulatory gene and the operator gene. Regulatory gene and operator gene these are the controlling mechanisms. They control whether the structural genes or the functional genes, whether they are to be switched on or not. Say, for example, genes get switched on only when there is a substance that has to be oxidized, that has to be utilized. When that is there, then only they become active and they produce mRNA, and thereby they release enzyme that digests or that breaks the food form or the energy source that is available to a bacterium or a cell, as the case may be. Now, these Jacob and Monod, while studying the effect of lactose, they proposed this operon. As this operon deals with lactose digestion or lactose hydrolysis, that's why it is known as lac operon, which consists of a group of closely packed genes. And these genes, as I already told you, structural genes and the control genes. Structural genes are 3. One is Z, Y, and A. Each code for a particular enzyme. All these together help in the hydrolysis of what we call the lactose, a sugar, a disaccharide. In the presence of lactase, of course, it is otherwise known as beta-galactosidase. The lactose breaks into galactose and glucose. This is what we call hydrolysis. So when sufficient amount of lactose is available, then only there is a need for the production of beta-galactosidase by the structural genes. So as I told you, there are 3 structural genes. One is Z, Y, A. And just very at the vicinity of this, there is an important gene that is, it also forms a part of what we call operon, that is operator gene. And the activity of the operator is in turn determined by another gene known as regulatory gene. Now this regulatory gene, it always active, and it produces what we call a mRNA or a protein, what we call a repressor. So when there is no need, like, when you go out, when it is not needed, generally you keep your door closed. When it is necessary, so that is always you keep your gate closed. It doesn't allow. When there is a necessity, when you want to go out or when you want to allow people to enter in, then only you open your gate. So here also the regulatory mechanism in the form of it produces a protein repressor that comes and binds with this operator gene. When it binds with the operator gene, then the transcription of this structural genes gets inhibited because they block That is, this repressor blocks the movement of polymerase, RNA polymerase enzyme. So RNA, this polymerase enzyme, movement of the polymerase enzyme gets blocked. So there is no synthesis of mRNA. So when lactose is absent, when sufficient lactose is present, then this lactose acts as an inducer. Say, for example, you feel like eating something. When you get, a pleasant smell of foodstuffs, generally your salivation gets enhanced and you feel like rushing for the food. Now here, when that lactose is present in the medium where this bacteria is being grown, then this lactose goes and binds with the repressor of the regulatory gene. Thereby it stops, so that it cannot come and occupy the operator. Thereby the polymerase enzyme can start its work, starting at what we call P gene, that is promoter area. Promoter area is the area where the transcription of the mRNA begins. So what I mean to say is, the production of the enzyme is in turn regulated by the food, that is the lactose that is present in the media. If it is present, then it counters the repressor activity and transcription for what we call transcription for the beta galactosidase, and that is Z, and the Y codes for what we call permease, that helps in the entry of the lactose into the cell, bacterial cell. Then adenylase is another transacetylase. This is another one. That this transacetylase is another enzyme. So transacetylase, your permease, and beta galactosidase, these three together work together in the hydrolysis of lactose. So the substrate or the lactose acting as an inducer. So that's why it is called an inducer system of operon operation. So what we learn from this is that just mere presence of genes is not sufficient. So their function, whether they are to be switched on or off, is decided by the medium, the presence of the nutritional source. They decide whether they are to be switched on or off. Thereby they'll be saving a lot of energy. So this is what we call lac operon. As it deals with the lactose hydrolysis, that's why it's known as lac operon. And this, because the controls, that is regulatory gene and the operator gene, so they form under what we call control apparatus or control category, which in turn determine the switching on of the lac operon. Of course, based on the substrate, there are a number of, for example, if the tryptophan is the substrate, then tryptophan operon is there. So like a number of operons are there based on the substrate that is available for digestion, or which is the nutritional source. So tryptophan operon is another another operon. Here it differs from this, that is lactose. Here the tryptophan itself becomes what we call that is not induced. Here the lactose was inducer, but here the tryptophan itself acts as a repressor. So when sufficient amount of tryptophan is there in the medium, then there is no need for the tryptophan synthesis. So here this is the synthesis operon, where that was an hydrolysis operon. So one is inducer, the other is what we call a repressor type of operons. Now this is about the structure of the gene and the operon concept. Now we move to the another important and the most important one. See, this operon concept may be asked in your exam. Operon, simply operon, maybe under short notes, or it can also appear under essay type. Now we move to another important topic, that is biotechnology. So now this biotechnology, what do you mean by this biotechnology? So the name itself gives you an idea, the meaning of what we call biotechnology. So the biotechnology, this is a new branch, a nascent branch, a recent branch of biology. As the term itself indicates you, bio, living, technology, application of engineering aspects to the biology gives rise to what we call biotechnology. So here, biotechnology is a product of interaction between biology and technology. It has resulted with the interaction between biology and technology, or where in biology, application of the engineering principle or technological principle leads to what we call the concept of biotechnology. Now this biotechnology, this biotechnology is a new branch, and it primarily consists of what we call, it led to what we call genetic engineering and the tissue culture. The biotechnology led to, or biotechnology, important components of biotechnology are your recombinant DNA technology or genetic engineering, and the other is tissue culture. Now this biotechnology is, as a principle, as a term, it may be new for us, but its roots are there in our civilization. We are using these biological organisms from times immemorial. We are habituated to make curd, we're habituated to make bread, we're habituated to make wine, alcohols. Now, these are the byproducts of microorganisms. These are the byproducts of what we call microorganisms. So now, this biotechnology, it is nothing but, according to what we call European Federation of Biotechnology, according to them, the definition of the biotechnology goes like this. It is the integrated use of biochemistry, microbiology, and engineering sciences in order to obtain technological application of microorganisms, cultured cells, or their parts. So here it is a new branch wherein the principles of microbiology, the principles of biochemistry, and the principles of engineering are combined together to exploit microorganisms or their cell contents on a large scale, that is on an industrial scale, to produce novel products in the form of vitamins, in the form of enzymes, in the form of antibiotics, on a large scale for the human welfare. This is the definition given by European Union of Biotechnology. So here, what we do, so it uses the, this biotechnology uses the principles of science and principles and properties of what we call microorganisms, or exploit the cells or their cell constituents at the industrial level for generating useful products that are essential for the human welfare. They should be for the welfare of the humankind. Now, say the basic difference. Of course, I told you microorganisms are in use, but here in biotechnology, not normal microorganisms. If you happen to change the chemistry, that is in terms of its genetics. That is, if you bring any change in its DNA or RNA, you're altering their gene chemistry. You're altering the genetic material, that is DNA and RNA. Then it becomes what we call altered. And if you are able to join the genetic material from two different sources, and if it is being introduced into a new cell, and that new cell having DNA from two different sources and combined together, gives rise to what we call a recombinant DNA. Recombinant DNA. So here what is happening? You are getting DNA fragment from one source, and you are joining this into an organism's or host DNA, and thereby making a recombinant DNA or a chimeric DNA or a hybrid DNA. Why it is hybrid? Its DNA is, an organism has its own DNA. Now you are adding, by using various modern techniques, you are splicing, you are cutting a fragment of the DNA in which you are interested, and you are bringing it, that, and you are joining to the host DNA. Thereby, you are giving it, it is giving rise to a hybrid molecule of DNA. That hybrid molecule is nothing but our recombinant DNA. Now, this recombinant DNA is being exploited nowadays to solve many, many problems. You'll be wonder, in near future, many unknown things may happen and your life becomes very, very easy. Now, we'll proceed one by one. So now, this principles of DNA technology. Now, here there are various steps. There are important steps are there. What we need in DNA technologies, we need what we call, I told you, you bring a fragment of DNA or a gene in which you are interested. See, before that, let us discuss about our various various hybridization experiments conducted by various people in plants and also in what we call in cattle. See, when you speak of this combination or bringing the desirable genes into one individual, that is, you go for what we call crossing. You select parents with desirable characters, because no parent has all the desirable characters. Now your aim is to get as many desirable characters as possible into a given individual. So for that, you select one parent as a male, the other as a female parent, you cross them. As a result of that, you get F1 generation, that is hybrid, which has the features of both parents. And in most of the cases, it is superior to its parents. That what we call heterosis, wherein the F1 or the hybrid is superior to its parents, maybe in various characteristics. So here, in traditional crossing experiments, in traditional breeding experiments, plant breeding, what we call, in breeding experiments, what do? You select two parents and you cross that. And you go on repeating that. Say, you'll be wonder if I say, for the production of one, a high yielding variety, a hybrid, scientists have to wait for 10 to 15 years. Such a tedious work they do. Day and night they work for the production of one superior hybrid. So here, you're crossing. So is the case with the cattle also. You take two cattle and you go for what we call artificial insemination, and thereby you get a progeny which is superior. Now, it is because of the crossing or the breeding experiments in plants, we could achieve what we call green revolution. This green revolution was because of breeding experiments. Whenever we speak of this green revolution, who comes to your mind? It is Norman Borlaug. Norman Borlaug, and of course, M.S. Swaminathan from India and others, they have tirelessly worked and ultimately succeeded in producing high-yielding varieties, first in wheat, of course, later on in rice, sugarcane, and this is going on. Almost all plants where high-yielding varieties are now available. Now, say in 1960s, we used to face a lot of famines, a lot of starvation deaths because of non-availability of foodstuffs. Now, it is because of the brilliant work done by plant breeders, high-yielding varieties came into existence, and by using them, we have exploited that. Now we have achieved, because of the green revolution, we have achieved what we call food self-sufficiency. Now our godowns are overflowing. It is because of the green revolution. What are the important components of green revolution? It was possible because of use of high-yielding varieties. What was the second component? Use of chemical fertilizers. Third was irrigation facilities. Fourth was use of insecticides and pesticides. Then this is nothing but plant protection. Then your harvest, your storage, and the marketing facilities. All these in turn led to what we call green revolution, and thereby we could achieve the self-sufficiency. Now here, what I want to stress here the breeder selects two parents and crosses. Now while doing so, I told you, no plant has all desirable features. Now while crossing, what happens? Along with desirable features, even undesirable features do enter. That is the danger. So while crossing, along with the desirable features, even undesirable features do come and get inherited. But that shortcoming, of course, another, our white revolution, wherein breeding in cattle. It is because of breeding in cattle, we could achieve what we call white revolution. Now, we are proud to say that now India ranks first in terms of milk production. This was achieved because of the breeding experiments done by our veterinary scientists. So here, similar to plant breeding and animal breeding, so when you breed, in addition to your desirable features, even undesirable features do get transmitted. Now, to overcome these undesirable features, now the technology of this, the manipulation of the DNA, or the manipulation of the DNA at the gene level by using what we call biological scissors. So here what you do, you know, the gene is nothing but DNA. And that DNA has genes like beads on a string. Now each gene, is nothing but a fragment, a part of the DNA, a fragment of the DNA that has nucleotides sufficient to code for a polypeptide chain or an mRNA. That is our gene. So now here what you do is, if you want to transfer, here you transfer not all genes. While doing experiments, hybridization experiments, it is not possible for you to select. You are crossing, so there is every possibility for the transfer of all the genes, desirable and undesirable. Here, you can select, you can mark, you can select a desirable gene, you cut it. Now, it was in 1970s, it is because of the brilliant work of Nathans, Cohen, and other Boyer, etc., the discovery of what we call biological scissors otherwise known as endonucleases. These endonucleases help us to cut the DNA, to cut the DNA or that particular gene, and this gene will be transferred into a plasmid. You know plasmid? Bacteria, we have bacterial chromosome. Apart from bacterial genome, we have small circular self-replicating DNA rings. They are called plasmids. Plasmids do exist. Their number varies from species to species, ranging from 2 to hundreds. Now, these plasmids, they have certain specific genes. They have the ability to replicate self. In addition to that, they also have certain important genes in their genome. So here, because bacteria multiply very fast, binary fission, divide very fast. Within 30 minutes, a bacterium, if you start with a bacterium, it becomes 2, then 4, 8, like this. Within 24 hours, their number reaches to lakhs. So within 2 or 3 days, their number grow up to what we call millions. Now here what you do is, so when million cells are there, means your number of plasmids also will be almost based on the number. If there are 10, of course, multiply the number with 10. So such a large number of plasmid population will be there available to you. So now here what you do is, by you cut the gene of your choice and you transfer that gene into the plasmid. You break by using various techniques. For example, various these bacterial, the cell wall, it has to be digested. Then the plasma membrane has to be digested. Then comes, by squeezing and other techniques, you can extract the plasmid and you cut the plasmid, you break the ring and you join the gene which you have collected from a given source. And the joining is helped by another set of enzymes known as ligase. So when the gene of your choice, you have joined with the plasmid DNA, so that plasmid DNA with the host DNA, it gets integrated because you have cut the DNAs by using what we call this endonucleases. They cut at the palindrome, sequences. Palindrome is nothing but, if you read from left to right, right to left, same. For example, Malayalam, same. Civic, same. So they cut the DNA or the nucleotides at a right place, and thereby these, what we call the open ends, they are called sticky ends, and there you join the DNA which you have selected, and thereby you create what we call the recombinant DNA, or that plasmid becomes a recombinant plasmid. Then what you do, you transfer this again into a bacterium, a bacterial cell, because you have the gene which you have incorporated. They got incorporated into this plasmid. So once it got incorporated, there is no scope for its loss. So when it multiplies, when it replicates, along with its genome, even the new gene which you have added also multiplies. And when it expresses, even the gene also expresses, and it expresses in the form of production of novel proteins. See, for example, by using this, we have achieved a number of novel products or proteins. For example, this DNA technology has been exploited in a number of areas. For example, in agriculture, in industry, and also in medicine. We have achieved, because in agriculture, I told you in what we call green revolution, of course, on one side it is very good, but the other side, the input costs have spiraling effect, because you're using pesticides, you're using high yielding varieties, you are going for multiple cropping, crop after the crop, thereby the soils are losing its supporting nutrients, they're getting lost. So the fertile soils are becoming barren, infertile. And the other side is, by excessive use of these pesticides, insecticides, of course, you are controlling the population of insects, thereby you are getting good yields. But the other thing is, these insecticides and pesticides, they are drained into your water bodies. And whatever the organisms, fish and other water living organisms, they are getting infected. They are exposed to what we call cancerous growths. Even human beings, if you take your grapes and other fruits, you can see, even vegetables also, on vegetables and fruits you will see the remnants of pesticides. If you do not wash them thoroughly, if you eat them in due course, you are liable for cancers. So they are spoiling your environment, they are spoiling your water bodies, they are spoiling your water living animals, human beings. So to counter this, to reduce this, we should go for exploiting the microorganisms. So here what you do, you transfer a gene that gives resistance, that kills a given pest. Say, for example, you heard about what we call BT. BT means Bacillus thuringiensis. The gene from Bacillus thuringiensis was transmitted to cotton. Now this Bacillus thuringiensis, it secretes, as a protection, it secretes certain proteins. They are known as proteins, crystal proteins, that are poisonous. Any organism consumes them, suffers. We discuss only about its effect on insects. The the insects that feed on leaves of cotton, they squeeze the entire foliage. Lot of loss for the farmer. Now their population can be checked by transferring this BT gene into cotton. Now, when it reaches cotton, when the insect feeds, when its insectal larva feeds on the leaves, it digests the cry proteins that are transferred. We transfer this cry gene from Bacillus thuringiensis into crop. So the crop cells or the leaf cells, they have these proteins. When insect swallows, feeds, in its gut, the inactive form of cry genes, they get activated because the pH of the gut is ideal for the cry proteins to become active, thereby they kill the insect. So you are not adding anything, just you have transferred the crystal producing gene from the Bacillus thuringiensis into your cotton. So brinjal also, of course, BT brinjal is there, BT cotton is there now, BT tomatoes are there, into a number of crop plants or food plants or vegetable crops, this BT gene has been transferred. This is one. Now another example is, for example, in case of tobacco. there is what we call a roundworm, nematode. Nematodes are there in the soil. They infect the root system of the tobacco. They form a number of galls on the root system. Thereby, the crop, not only the quality, the quantity also gets affected. So here, from Agrobacterium tumefaciens, from Agrobacterium TI plasmid, by using TI plasmid, a gene that silences RNA ribonucleic acid interference, the name of the gene, that was transmitted into tobacco. Now, when this helminth feeds on the root juice, now that Agrobacterium tumefaciens RNA silencing gene enters into the nematode, and thereby it silences its RNA. Thereby, the insect or the helminth dies. This is another example in crops. Not only that, cold stress, your saline stress or heat stress, they are being countered. We are able to, stress relieving, stress resistant, cold resistant, heat resistant varieties are being released. To them, your novel genes are being injected into those crop plants. So thereby you can enhance your crop production. Now in tomato also, there is a flavor saver, a variety wherein, ripening of the fruits is by ethylene. A gene has been introduced into tomato. It checks the precursor of ethylene. No production of ethylene, so thereby you can prolong its shelf life. They go for what we call slow maturation, so that you can transport your product to longer distances. So thereby you are enhancing the shelf life of your tobacco. Tomato. So these are some of the examples where the biotechnology or genetic engineering, in terms of transferring the novel genes or the genes of your importance into the crop plants, thereby you have saved, you have enhanced the production, and you have protected your crop. Now, if you move to industry, in industry also we have achieved a lot of success. Now in industry, say for example, food industry, for example, we know our yeast, now it now what yeast does, when sugar is there, it converts. Partial oxidation leads to what we call carbon dioxide and ethanol. Of course, that is being used. Now, into yeast, cellulose digesting enzymes are introduced. So they not only digest your glucose, they also developed the ability to digest even your grass. Here the cell wall is made up of cellulose. Generally cellulose is very hard material that can't be digested. So by producing, injecting novel genes that are capable of digesting your cellulose into a yeast, you can you can digest. So the waste material can be used profitably. Your all logs, all heavy unwanted material, plant material, that can be profitably converted. You can convert them either into glucose, or they can be converted into what we call starch and other things. And also the other thing is this biodigestion, it can solve your pollution problem, but also they will help you to generate what we call biofuels. You can also generate, you can check, on one hand you can check the pollution, the other hand you can produce biofuels. Now, the other thing, say for example, a lot of work has been done in medicine, health. Now in health, here, both in digestion and therapeutics, in both the areas, these have been exploited on a large scale. See, diagnosis, in diagnosis, generally, we go for what we call urine examination, blood tests, etc. Before a doctor gives you a prescription, he asks you to go to get various tests. Now, even these tests are cumbersome, expensive, and even they are not accurate. See, for example, now let us speak about what we call COVID. If you experience what we call temperature for 1 or 2 days and cough, etc., you get scared. Is it because of COVID? And of course, in yesteryears, years, we have the problem of what we call HIV. We don't know. So for them, you have to wait. For AIDS, it takes months for the appearance of symptoms. In COVID, etc., it may take a week or so for the appearance of symptoms. By that time, the population of that particular pathogen will be at its threshold. Then it becomes very difficult for us to counter them, to kill them. So early diagnosis is possible by using this recombinant technology, by using innovative methods such as our, say for example, RT-PCR test, polymerase chain reaction. A small, say if a pathogen enters your body, means naturally your body reacts and produces an antibody. Here, even the presence of very few number of antibodies is sufficient. By using these new techniques, even the within 1 day or 2 days after the entry of the pathogen, maybe HIV or your COVID, you by analyzing, by doing tests for your antigen or for the antibody, we can say that whether that particular individual is affected by that particular pathogen or not. So, and another, ELISA test, this enzyme-linked immunosorbent assay. This is nothing but your antigen antibody. So this is also a perfect testing. So all these help you to arrive at a conclusion, an accurate diagnosis. Then comes to your therapeutics, of course. Say nowadays, monoclonal antibodies, now even for our treatment of your COVID also. So here monoclonal antibody, if a pathogen enters, your body reacts and produces an antibody. Now here by using this technology, by you extract that antibody, and by using this PCR, polymerase chain reaction, you produce n number of copies, billions of copies. So you generate the cells in millions and billions, and they, after purification and formulation, etc., they can be used. So for antibodies, and also, for example, in biofortification, say nowadays, yellow rice, this rice, golden rice. Golden rice is rice which is fortified with vitamin A. In addition to vitamin, calcium, zinc, iron, your thiamine, riboflavin, niacin, pantothenate, almost all B complex are being added to the rice. So here then you need not go for vitamin tablets, multivitamin. If you take that rice, fortified rice, that gives you good health. This is because of exploitation of microorganisms or our rDNA technology. This is possible because of this. Now, insulin, I told you, big story of insulin. In yesteryears, for 30 or 40 years back, what used to be the treatment for the insulin? Now, scientists used to extract insulin from the slaughterhouses by killing a pig or a cattle. From that pancreas, they used to extract. And that after purification, they used to give. But of course, because you are giving the insulin extracted from the animals. Your body and the body of the animal differs. So there are immunological problems. But here, by using this biotechnology or RDNA technology, scientists have successfully excised, separated the human, insulin producing gene, and that was introduced into an E. coli. And now by culturing this E. coli, you can produce more and more amount of insulin in pure form, and that is available everywhere now. Not only insulin, your growth promoting hormone, human growth hormone is there, interferons are there, your antibodies are there, your vitamins are there. So lot number of novel products are being produced on an industrial scale, on a large scale. And because of its production on large scale, the cost effectiveness has come down. Now, when early to 2020, when we faced, our COVID, then the cost of our Covishield or Covaxin was very high. Now, because of use of technology, and because of the intervention by the government of India, now we are able to produce on a large scale. Not only we could give sufficient doses to our population, almost 80%. First dose, of course, almost 70% of the population, and more than 60% receive second dose. Now, not only Indian population, but we are in a position to export that. This is because of use of technology, RDA technology. Now we are able to produce vaccines. Vaccines for COVID and vaccines for many other diseases. We have polio. We have rabies vaccine. We have vaccines for number of your diphtheria, pertussis, tetanus. Number of vaccines are available. Your typhoid. Number of vaccines are now available. In almost all the areas, they exploit the microorganisms and they alter the genetic material of the microorganism. And this chimeric DNA is grown on artificial medium by using, bioreactors, big vessels, and under controlled, pressure and temperature. The cultures are being added, and used cultures are being drained, and thereby the production will be in large amounts. So this is how the biotechnology has been used profitably. Now, lastly, I'll tell you, say, it is because of use of biotechnology and tissue culture. Now, the days are not far. Now, what we are doing, if a person dies, brain dead, then doctors advise you to donate organ donation. Thereby, your heart, your kidney, your liver, etc., are transported, and they are being given to the patients in need. Now, the days are not far wherein embryonic cells of your heart, embryonic cells of your liver, embryonic cells of your kidney, and all other parts, they will be grown in the laboratory conditions, and your full tissue is available, or the organ is available, that can be transferred to the patients in need. Now, here there is no scope for what we call immunological disturbances. So this is how the biotechnology or the genetic engineering are now being exploited on a large scale for betterment of the human society, for the welfare of the society. Now, with this, I'll conclude my lecture, and thank you very much for your participation, and wish you all the best. Now, my piece of advice for you is, because of time constraints, we have to rush through, and I advise you, better you go through intermediate books. There the basics are available. Then you read the material that is given by university, and you combine that, and you can make a good presentation in the forthcoming examinations. Wish you all the best. Now with this, we have uh, we are concluding the teaching, online teaching of what we call uh, uh, the cell biology and genetics. If you have any query, I'll answer. If there is no query, and I'll call it a day. Thank you.